Thank you so much again, Reinhard, for inviting me. What I'd like to do in my 15 minutes is to give you two sketches. They're very sketchy. The first one is showing how we think about the life cycle of a digital twin. And the second one will be more specific about AML digital twin for acute myeloid leukemia. As I mentioned when I introduced myself, we have started a project to develop a prototype for the AML digital twin. So I completely share the optimism of several in the room, Tom, others, that we can get somewhere. It will never be perfect, but if we can do a little bit better than standard of care, then that's already a great step forward. So first, I'd like to start with the digital twin life cycle. We have a patient, and we generate multi-scale and multimodal data from the patient. When the patient comes into the clinic, various kinds of data. It depends on the tumor type. For AML, it's going to be some specific data types: flow cytometry, cytogenetics, mutation panels, so on. Clinical data, of course. Then we use this data for building models, training the models, or I should say calibrating the models to the data. And by the way, I should say that this figure that I'm reproducing is published in a commentary in Nature Medicine. Paul Macklin is a co-author, my partner in crime for this, and also several other collaborators in Stanford, the National Cancer Institute, Department of Energy. It's a group of us that conceptualized this view of the cancer patient digital twin. So the model is then used to explore digital twin predictions. So this could be disease trajectories, predictions of toxicity, adverse effects, responses to treatment. So it's a computational exploration. So these explorations have to be visualized. This is my terrible attempt to draw a laptop. This is a keyboard. It has to be visualized with some user interface in what we're calling a dashboard. So these predictions can be interactively explored by the doctor and the patient. So here's the doctor, here's the patient. They are working together. The patient is participating in the decision making to the extent that the patient wants to participate. Many patients feel like they just receive the recommendations, but they don't feel that this is a participatory process. And the digital twin will help enable this aspect of clinical care. And then individual care decisions will be made. Individual, emphasizing that every patient is unique at some level, and every decision must be tailored to that individual. Of course, people who actually do practice medicine will say, well, it's already that way. We already try to individualize the decisions. We do the best we can. But this is going to be supported by these computational models, which are constantly updated. So there is a continuous learning cycle where, as many patients are studied and treated with digital twin technology, we will have then cohorts of digital twins. We will even be able to create cohorts, virtual cohorts of digital twins. And with such capabilities, we will be able to do population studies. We will be able to advance basic science. So for developing new interventions, for example, understanding the mechanisms better. And we can even do virtual clinical trials or enrich cohorts for actual clinical trials. So there's a continuous learning loop. Continuous learning. As more and more patients are engaged in this, the digital twin technology will get better. But also, over multiple clinical encounters, even with one individual patient, the actual digital twin for that patient will also get better. It should get better because there's more data that is collected. So whatever calibration of the parameters is performed, it will be refined as more and more data are being used. So this can be used to adjust treatments. It can also be used for monitoring. Hope you can read my handwriting. Monitoring response to interventions, to treatments, therapies, and even track lifestyle modifications. So for example, another project we're working on is diabetes digital twin. This is where lifestyle modifications are very, very pertinent and consequential. So this is the life cycle of the digital twin. I'm sorry, I'm going to now go to here, and maybe you won't. I'll try to keep it here so you can see it. Sorry, Reinhard and Luis. I know you're in that zone. So now, AML, acute myeloid leukemia. Obviously, it's related to the immune system. It's a blood cancer. It's a hematological malignancy. There are multiple types of leukemia: ALL, CML, other blood disorders, MPN, and so on. We decided to focus on AML because it has, not only because it has dismal prognosis, but also because patients routinely get multiple measurements over time, relatively non-invasively. They're bone marrow biopsies and aspirates, which are quite invasive and intensely painful, but they do get, multiple such biopsies throughout the course of the disease progression. But they also routinely get peripheral blood draws, and that is also quite informative of disease, with information like CBC differential and various other panels like metabolic, renal, other kinds of panels that are informative of not only disease but also side effects of treatment. Because many patients, after they receive various chemo drugs or other interventions, they become, for example, cytopenic, pancytopenic, or neutropenic. So they don't have neutrophils. They become vulnerable to infection. They become thrombocytopenic. They don't have platelets. They don't have red blood cells. They can bleed. They need transfusions all the time. So patients get blood draws sometimes daily. So you can get data, very nice time series data, something you cannot get from solid tumors, right? From, if you imagine something like pancreatic cancer, brain cancer, it's very difficult to get multiple time point measurements unless it's imaging or maybe biopsies. But with blood disorders, you can get really good data. And so that's a good starting point for building a digital twin because we have this time series data. So again, here's the patient and the doctor. The patient, there are various kinds of data types. Maybe this box is too small. So there's clinical data, obviously I mentioned, from peripheral blood, from bone marrows. There is flow cytometry. This is routinely collected in all clinics. I mean, the actual assays may be different, what markers they use for flow cytometry. But generally, it's going to be the same for identifying the percent of blasts, which are the cancer cells, but also other cell types in the bone marrow with different markers. Flow cytometry, cytogenetics. So looking at large-scale rearrangements and aberrations, genomic aberrations, which can be used to stratify patients into different risk groups. There is molecular data, such as genetics, as I mentioned, genetic panel. So these are mutation panels. These are also very informative as for deciding which drugs might be effective. Immune profiling. This was mentioned before in one of the presentations. Cytokine profiling. Now, some of these things are not routinely done, but they can be. Of course, there's a cost associated with it. And even transcriptomics, like RNA-seq from the bone marrow, or single-cell RNA-seq, which can show you the different populations of cells, including the leukemic cells. And then there are also treatment response measurements. After the patient is treated, there are various measurements that can be made to look at response. So here's my laptop picture again. There is a dashboard that is used by the doctor and the patient to visualize the predictions of the digital twin, visualize the historical data, like a dashboard, and this is actually used at the Fred Hutch. I'm working with collaborators in Helsinki University Hospital in this project, as well as with the Fred Hutch Cancer Center. And they're already using a dashboard to visualize retrospectively, the disease progression. When the patient was diagnosed, what treatment was on a timeline. When the treatment started, when it finished, you can overlay various, side effects, anything. Cytopenia, shortness of breath, liver function, and so on. You can see historically what was going on. But right now, you cannot look into the future. But with the digital twin technology, we will be able to use the dashboard to also look into the future, make predictions, predict drug response, for example. And we will be able to predict adverse events. As I mentioned, things like cytopenia, for example, which is very important. The actual engine of the digital twin is going to involve ensemble type of modeling. Not just one model, many types of models, depending on what it is that we want to predict. It will include multi-cellular modeling. This will be typically agent-based modeling that will explicitly model stem cells, hematopoietic stem cells, leukemic stem cells, progenitor cells, other cells such as lymphocytes, particularly in the context of allogeneic stem cell transplantation. This becomes very relevant because the immune system can exert a graft versus leukemia effect. So the T cells can actually kill the cancer cells. Of course, they can also cause graft versus host disease. So it's two sides of the same coin, but this multi-cell model will try to capture these effects with measurements, obviously based on measurements and tuned to these measurements. This model, there will also be a genetic regulatory network model, GRN, which will model on the molecular level the aberrations of genetic regulatory networks in AML cells, in leukemic blasts. And these models will be used to generate features. And then these features will be input into a machine learning phase of the model. So you have features from multi-cell modeling, from genetic regulatory network modeling, and there may be multiple models even within these boxes, but they will all be used to generate features. And this will result in personalized models. These models, the machine learning, will be used to visualize these predictions in the dashboard. There is a lot of public data. AML is also quite a good cancer to study because there are many large data sets for functional screening, drug screening, genomic data sets, cell line data sets. I'm not going to list all of them, but there are quite many. So from these public data sets, I'm just going to draw this kind of a database. So you have drug screens, functional screening, things like with CRISPR screening, multi-omics data sets. There is, for example, a big data set called Beat AML, which does genomic transcriptomic profiling together with drug screening, ex vivo. So they get the primary patient cells, do the screening ex vivo, have the results, you have the data. Also, of course, clinical trials. And so all of these data can be used to generate so-called knowledge graphs. Knowledge graphs. These knowledge graphs, and we've already done a lot of this work in my lab, so they're based on general knowledge graphs that are not disease specific, but once you start overlaying these data on the general knowledge graphs, they become disease specific knowledge graphs, essentially AML knowledge graphs. These knowledge graphs can be then used to engineer features that also go into the machine learning stage. They also can be used for creating the gene regulatory network models. So this machine learning box essentially integrates features from all of these models, mechanistic models, network models, knowledge graphs, to make individualized predictions. I should have also drawn an arrow here that shows model integration. I'm finished. You didn't have to say, just got up and that's enough. Yes, we're well trained. So all these data are of course used to instantiate the models and to calibrate the models as I showed over here. So this is a 5-year project. We just started this year on this project, but we've done a lot of preliminary work on drug target prediction using these approaches. We're now starting to work on mechanistic models to predict disease progression, even in the absence of therapy. Okay, I'm going to stop there and we can have a discussion. Thank you. Thank you.
Is this going to require a doctor to have any intimate knowledge of the model, or is it more black boxy for them and the patient to where they just click or, I don't know, enter in various variables or potential lifestyle changes or whatever it is that's going into it that then would inform that decision?
Yes, at some level it will be a black box. The clinician does not need to understand the intricacies of this. And in fact, the dashboard will be designed for the clinician and for the patient separately. So the view of the patient may be different than the view of the clinician, although the clinician can turn the screen and show the patient, well, here's the prediction that we see. The clinician already has a pretty deep understanding. They just may not need to know the details of the mechanistic models necessarily, unless they want to. But they already have an understanding of how these measurements, like flow cytometry, cytogenetics, and mutations, how that leads to the predictions and ultimately their decision making. I mean, there are already guidelines, obviously, plus most clinicians have their own experience from them and their colleagues for making these decisions. But that's actually a general problem in the field, is that the people retire, their new people come in, and the knowledge doesn't always get transferred. And this kind of the digital twin will help to democratize this capability. There are also some cancer centers that are, top-notch centers, MD Anderson, Sloan Kettering, Fred Hutch, but then there are other cancer centers that may not have the same resources or the same experience as others. So a technology like this could help democratize this and improve outcomes for patients everywhere, not just in the top clinics. And as I said, the patient will also be able to participate in the decision-making process. They will feel that they're part of the process of making the decision, and they're not just on the receiving end. That's very important.
Can I ask a follow-up question then about validation and what kind of inputs you're getting back about the success of the model?
Well, the success will have to be, it was mentioned in one of the earlier discussions that we have to have clinical trials to actually use this as a software, as a medical device, get it FDA approved, and introduce this into the clinic and show in a randomized clinical trial that patients who have the benefit of using the digital twin with their doctor have better outcomes. Now, what outcomes are we talking about? That's to be defined. What are the endpoints? It could be the traditional ones like overall survival, disease-free survival, and so on, but it could also be patient-reported outcomes. What is the quality of the care that the patient receives? How satisfied is the patient with the entire process? Many patients get very frustrated, and this also leads, this is an important outcome, that their quality of life suffers because of that. So that would have to be designed in the future. That would be a validation. If you can achieve better outcomes than standard of care, if you have a two-arm trial, one is standard of care, one is standard of care plus the digital twin. It's not a replacement like the dermatologist story. It's basically an add-on. The clinician still is in charge, still makes the ultimate decision, but this is a clinical decision support tool. I emphasize the word support. It is not replacing the clinician and saying, well, no, you step aside, now I make the decision for you. That's not at all what it is. Paul. Oh, I'm sorry, Tom.
Well, my comment will end with I defer to Tom. And actually, they're both Toms, Nebraska Tom and Texas Tom. But I think it's very useful about the idea of both patient and clinician facing dashboards is to kind of tap into Kristen Swanson's notion of a virtual control, right? That you want to run virtual experiments on your patient to kind of weigh the benefit and risk of a lot of treatment varieties and different treatment plans. And ultimately, you need to be able to visualize how well this does compare to if we had just left them alone for quality of life and for the toxicities of that treatment. And so having this dashboard be able to say, here's how you are today, here's how we think you would go without a treatment for your virtual control, and here's how we think you're going to go on our best and the treatment we selected together. Do you feel that this improvement is worth the toxicities you're about to undergo? And then on the flip side, it's quality of care monitoring, saying, here's where we thought you were going to go. Here's where you thought your treatment control was. And if they match, that's great. That means everything's hunky-dory. And if not, that means something's changed in this patient. We better get down and figure this out. This is time to do some more diagnostics because your predictions did diverge. So lack of prediction is actually also very predictive from a prognostic point of view. That model failures are an indicator that something's changed and your assumptions are no longer valid. It's time to go do some digging. And so it's not actually a failure of a model in that sense. It's just, it tells you it's time to do some hard work. And now I defer to Tom, both Toms, but Texas Tom.
For the record, I'd like to say that I actually live in Austin, Texas. I don't live in Texas. I live in Austin.
Good. Yeah.
So fantastic. I really enjoyed your presentation. It's very easy to see how you got a 5-year R01, which is no small accomplishment. Well done. Well done.
Thank you.
So keep that in mind when I ask this question, and don't try to read past it or through the lines. So it'd be great if you could unpack for me both philosophically and technically in that box there where it says machine learning and then it says personalized model underneath there. So tell me seriously, from a philosophical point of view, how you can land on a personalized model through a machine learning component, and then maybe technically tell me a little bit more about how that's done. It'd be great.
Yeah. Thank you, Tom, for your question. I lived in Houston, Texas, so I take your point. I think that it's personalized because we can of course argue about the nomenclature, but the reason I called it personalized is because it's using patient-specific data, mutations, flow cytometry, expression profiling from the patient. So the model is calibrated to that patient. And as there's more data gathered over time, the model is recalibrated. So this is what makes it more of a digital twin model. It's not a static model. It's a dynamically updated model. So that's why I called it personalized. The machine learning stage it could be anything. It could be random forest regression, lasso regression, neural network. That's not relevant. We don't even know what it's going to be at this stage. We're not focusing so much on that, but we're focusing more on whether these features are going to be informative, whether they will have predictive capacity. So we are hoping that we already know that the knowledge graphs, when we engineer the features from the knowledge graphs in a disease-specific manner, they are predictive of drug response specifically. We know that if a patient has these particular molecular characteristics, the patient is more likely to respond to this or that drug. We know that because we can test it with large data sets. However, we don't know yet whether these mechanistic models of gene regulatory networks or multi-cell models will be able to also generate additional features to make these predictions even better. Or the other kinds of predictions we want to make is not necessarily drug response, but disease progression. AML develops really rapidly. The doubling time is short. It's like 11 days or something for blasts. So we want to be able to make predictions even like 1 week or 2 weeks into the future. So this is also why AML is a good starting point compared to much slower developing tumors. We would like to be able to use these mechanistic models. They may be ODE models that model the bone marrow, the niche, where you have the stem cells, the leukemic stem cells, they compete for that niche, they get ejected. So there are already some published models, ODE models for the hematopoiesis and leukemogenesis. There are also some published models on gene regulatory networks for AML. So we already have some starting point. We don't have to start from scratch. But the hope is that the parameters of these models, or maybe the outputs or whatever, some features from these models will be also predictive, in addition to what we're seeing with knowledge graphs. So this is still a question mark for us to explore. I hope I answered your question at some level. Thank you, Tom. Okay.
I got a little stuck on the first sketch, actually. I'm trying to understand how the ensemble of digital twins allows you to build virtual clinical trials. It seems like you would need to understand the effects on each digital twin already in order to do that.
Right. Yes. I mean, it's sort of, I see what you mean, because the whole notion of a digital twin is that it's sort of an N of one. And a clinical trial, you have a cohort. These are like different things. But to the extent that this could be useful for trying to designing randomized clinical trials, you could try to design a clinical trial in a computer, right? If you wanted to design an actual clinical trial, which I think is something that's going to stay for a while, the FDA, this is not going to change immediately. We're still going to be stuck with randomized clinical trials with all of the drawbacks. But if you wanted to do that, you'd be able to do that in a computer. If you really do have a good population of digital twins, which are diverse, which is one of the challenges in clinical trial design. How do you control for that diversity? What do you control for? Do you control for age, for sex, for other kinds of comorbidities? So we will be able to explore that in a computer to help us design better clinical trials. I think that's the point, but maybe, Paul, maybe you want to also partly answer this question since we thought of this together.
I don't want to talk over their fellow Husker too much, but it's interesting, right? Because you can train the digital twin to maybe some specific assumptions and concentrations of the drugs, and then use the virtual trial to test different intervention strategies, dose timings, dose escalation, skip doses. So you could still explore strategies with a virtual trial, even if you had a bunch of good individual digital twins. So I think that's one thing. The other thing is, there may still, I think, be a lack of access, that some clinics may not be able to implement digital twins because they don't have, the imaging modalities, they don't have the data sampling, capabilities. And so, the virtual cohorts of digital twins could still be good for designing trials to say, now that we know more from the digital twins, can we provide better generalized treatment strategies that work across the board, or account for the fact that maybe there was too stochastic to come up with an individual prediction, but we can come up with a better probabilistic treatment that says, even for this cohort that's, 30% of the time they do this, 75% they do that, but this strategy seems to help shift that balance to maybe 20/80 or 10/90, and a virtual cohort and virtual trial, I think, would be very useful for that kind of exploratory work.
Indeed.
To designing better treatment strategies for when you can't employ the digital twins. And I want to get out of the way.
Thank you, Paul.
Can I ask maybe just a definition question? Because I know that Reinhard doesn't think that virtual patient cohorts are digital twins, but this sounds a lot like a virtual patient cohort, but might also just be the same thing as a digital twin?
Well, it's a cohort of digital twins.
What does that mean?
What does it mean? It means that you generate in a computer many virtual twins.
Right, but you do that in a virtual patient cohort.
Why would you?
No, you do that also when in a virtual patient cohort, there's trajectories of different patients and they're all individualized.
Okay.
It's the same.
Yeah, it sounds the same.
Reinhard, can you comment on why you think it's different?
Yeah, so maybe I can clarify the remark I made. It seems to me that if you actually match your digital twin population to an actual population, then I would call it a digital twin. But what is typically done, and we do the same thing, is you vary parameter settings, and that gives you a collection of patients that have no immediate connection to any actual patients. And that's where my comment comes from. I'm not saying that's not a good thing to do, but it's generally difficult. Right? You have an actual population. Let's say I want to do what Ilya says is I do a clinical trial, but I want to supplement the population with virtual patients, maybe to achieve an appropriate distribution, then I think it's really meaningful from the point of view of digital twinness if you actually have a way to pick parameters that match particular patients. That was my comment. Let me go to Tom. Tom makes sense.
So I think population PK models, right? We're simulating that the Certara and the pharmaceutical industry has been using this for a while. I think the advances that we're talking about here is that the pop PK models are sort of black box, right? And you use different distributions to generate your populations. And there are also actually databases that have very realistic characterizations of different populations from different countries and continents. But I think the opportunity for advance that we have here is that population PK looks at a couple of organs. And so here we can actually unfold that black box with molecular, multicellular level detail that will hopefully give us more insights into the mechanism of action of a drug, a potential treatment, or even the disease mechanism that we don't currently get from PK models.
Absolutely, and that's what here I wrote basic science. It will help advance our understanding and possibly even develop new interventions. That's right.
So I just wanted to give an example that we did a couple of years ago. So we've been doing virtual clinical trials, and we have very nonlinear hybrid multi-scale models that span from molecular all the way up to what we consider the whole host. And I can talk about that a little bit more for the disease TB. And there are 15 clinical trial vaccine candidates currently undergoing investigation because the current vaccine for TB is terrible. And so we had human data from a clinical trial, we had non-human primate data, and then we had our model. So we simulated our model and we tried to follow the protocols for both the vaccine trial that the monkeys did and the vaccine trial that the humans did. And we were able to predict lots of things and we could do it in a general sense where we weren't matching cohort, or we could match specifically to patients because, there was a very limited number of humans and a very limited number of monkeys. So we could do both, right? Because we could make one-to-one correspondence, and we can also create a ginormous virtual population of potential, I'll say, twins. And we were able to predict things like if we had done the model first, you wouldn't need the third boost. The third boost was a complete waste of time. At any scale, whether you look at the individual scale or whether you look at the population scale, the third boost showed no improvement. It was a waste of energy and patient time and cost. The other thing is that we looked at the parameter spaces, which I'm very much interested in, between the monkey and the human and the model. And believe it or not, the mechanisms that were underlying the dynamics of all three were consistent, but we could figure out where the differences in the parameter space would exist, which was actually quite satisfying to know that we're capturing the main dynamics at both scales, the population scale as well as the host scale. So I think that there is what Ilya is describing here is possible, the translation between all of these different scales. And I think we're already doing it in many circles. And it's the problem is getting anybody to do it a priori before all of this starts. And I can tell you this, that we work directly, we have funding from Bill Gates. And what we do is, not just vaccines, but we also do drug regimen prediction. And so they asked us to prioritize through an optimization protocol the top 10 drugs for TB, and what combination and perturbation can you give them to do the best response. and they were doing it in the mouse, and we were blind to their predictions. So we did it, and we made our prediction with our model, and they did it in the mice. And there were many similarities, but there were a few differences. They're like, well, you're not matching the mice in a few of those things. I'm like, well, you're not matching us. And who do you believe more? A mouse that doesn't actually have granulomas and have the same immune response, that we see in humans and non-human primates? Or do you believe my model, which has been validated at every scale and is hybrid and capturing what we see at all the different scales in the human and the monkey? So changing that mindset, they really want us to confirm their mouse models. But right now, in all diseases, the studies are done in drugs. The prediction is made in mice for drugs, and then they scale up. They use an allometric scaling to get to the human, and that's what they treat with. That is mind-blowing that they think that model is better than any model that we would create in a mechanistic way. But that is done every single day. And so we have to convince them that our models are better than their mouse. And by the way, that's true for both vaccines and drug treatment. And we wrote a paper a couple of years ago with a group in the UK about this for vaccine, that maybe there should be a little computer picture in between the mouse and the human that would, maybe feed some of the mouse data at least and see how the prediction goes. But I mean, I don't know, you guys, we have a long road ahead of us to get them away from thinking that mice are the be all and end all for every single disease system that exists. So sorry for my rant.
So I think this is a great ecosystem and disease process to show the scale of time series data necessary for the process. So I think that is you're to be commended for that this particular structure, because I think it's exactly a type of data collection ecosystem that we need to do for these other types of projects. I think some of the features associated with AML that allow you to do that, the fact that you can sample blood, that's actually great. I shared the same concerns that Tom from Austin has regarding the ML component. You alleviated that to a certain degree by talking about ML methods other than neural networks, because, the curse of dimensionality is going to get you on that part. But I think overall to be commended for that, this structure.
Thank you, Gary.
And also hashtag just saying mice for Denise.
Thank you.
Just one short comment. That's what you're doing. It's great. I mean, for a project on ML, I just want to step back and question the fact that we are all, I don't know, maybe I'm wrong, but we were thinking at the digital twin as a unified computer program, basically. And myself, in the past, I've been developing a model of the immune system, and I've been adding components like a gene regulatory network for specifying if a TH will differentiate here and there, and so on and so forth. Now, the question I want to raise is, why shouldn't we stay at the level of keeping models separated according to the origin of the data? Because after all, there are, we are one system, but certain part of our components don't really talk between each other. So why should we try to do such thing, which is, I think it's too much. It's beyond our capacity. And why don't we just keep it separated? The model, it will be not one model, but models. So a digital patient is a collection of models.
Yes.
This looks to me more realistic, and it fits actually that the two box you have drawn there, where you have different data and you create a different model for it.
Yes.
Then the problem of the integration will come after. And to me, it's more realistic to think to integrate few of those models than to think that you will integrate everything.
Yeah.
I've been working for 3 years on a model of diabetes, and we integrated 5 models. It has been a nightmare, and still it's a model of type 2 diabetes. It's not a model of cancer. It's not a model of infectious disease. It's not a model of health. It's not a model of nothing. It's just a model of type 2 diabetes, not even type 1. And it has been a huge effort.
Yeah. Thank you so much for your comment. if I may just say one last remark. I agree with you that we need different models. I think I made this point, but I want to re-emphasize it, that this is not just one model. These will be multiple models. Even in each of these boxes, it may be multiple models. But it's not always so clean as one data type, one model, because, for example, in the multicellular model, that model can be informed, for example, by flow cytometry data, but it also can be informed by RNA-seq data. So these two data types can tell us about the cell type fractions that we need or the initial conditions in the multi-scale model. So it's sort of not a one-to-one mapping, but it's maybe like many-to-one mapping or many-to-many mapping. But there needs to be some separation, and indeed, this integration can happen at the level of machine learning. This is why we conceptualized it in terms of integrating different features from different models, even just statistical models based on knowledge graphs, and then making the overall prediction based on this aggregation of features using, could be even simple approaches, such as random forest regression or other kinds of well-studied approaches in machine learning. I don't think that's really the essential component here. So thank you for your comment.
All right. Thank you. We'll have to leave it at that.
Thanks.